So it's my pleasure to introduce Tom Diethe. So Tom did a PhD in UCL and then a postdoc in Bristol, and then moved into industry working for Amazon in Cambridge. So moved over to Cambridge at that point. And then after Amazon research, moved into AstraZeneca. And so has a good kind of insight of both the sort of academic track of research and the industry in tech, in Amazon research, the sort of ML lab there led by Neil Lawrence, and also in the sort of pharma AI. So so maybe you can grab him afterwards and talk about all those different experiences. Find out which is the best choice, I guess. He'll say AstraZeneca. So thanks, Tom, for coming, and over to you.
Sure thing. Hear me okay? Great. So thanks, Magnus, for the opportunity, and welcome all. Hope you're not too bleary-eyed. Let's see if this works, and I'll start this. Great. So I guess many of you will have heard of AstraZeneca through, from COVID or maybe before that, but brief bit of history, which it was actually two different companies, Astra and Zeneca, that merged in 1991, British part and a Swedish part. So it's still an Anglo-Swedish company with dual headquarters. Well, the main headquarters are now in Cambridge in the UK. But I think that it's quite interesting because maybe different. It's certainly different to other places where I work because it has a bit of a Swedish sort of culture that pervades the company as well. My role is that I head up the Centre for Artificial Intelligence, and that's part of what we call biopharmaceuticals in R&D. Essentially, that's everything that's not oncology. So that's covering cardiovascular, renal, respiratory, immunology, vaccines, etc. And yeah, so I titled this Machine Learning Applications in Drug Development. I've given up personally trying to discriminate between what's machine learning and AI at this point in time. So, Centre for AI, we do machine learning basically. And in AstraZeneca, one of the core principles really is about following the science, and that means for every drug that gets developed, it's really key that the sort of the paper trail that shows that all of the scientific work is really backing that, and that extends backwards into data and AI as well. So it's really about being extremely rigorous about the science that we do. And just some numbers, some quite scary numbers really. Globally, 2 billion people have some kind of chronic disease that falls into this biopharmaceuticals area. Top 5 causes of death: cardiovascular disease, COPD, chronic kidney disease, and diabetes by 2040. And 1 in 3 adults now have more than one chronic disease. And the economic burden, obviously big numbers in this industry, $22 trillion. So that's from chronic diseases and COVID. And these are the kind of molecules that we work with. So you might think of the traditional ones being those ones in the top left, so small molecule drugs, antibodies, so monoclonal antibodies, and other sort of therapeutic proteins. But increasingly, the portfolio is expanding into all these different modalities. So antibody drug conjugates, that's where you're using an antibody as a carrier molecule and attaching the actual drug part to the antibody itself. Bispecific antibodies, where you're sort of like pulling apart an antibody and using 2 ends of it to do something. little fragments. Cell therapies is a huge and a new area. Peptide-based, so those are just small chains of amino acids. DNA-based drugs, RNA, et cetera, this goes on. So this is kind of an interesting challenge. If you think, maybe you think AlphaFold solved protein engineering, well, it hasn't. And it certainly hasn't solved all of these problems in all of these modalities. And, again, to what we're trying to do, I think traditional medicine has been a lot about tackling symptoms, and increasingly, we're really trying to, understand more about the underlying target biology to head much more towards treating the actual causes of disease. And in order to do that, you have to understand causality as well. So causality is a big deal for what we do. Data science and AI really are at the center of things now. So I mentioned about disease understanding. I've mentioned about next generation therapeutics. We're pioneering new approaches in the clinic itself. So that's with maybe with digital technologies more than it is with machine learning, but increasingly things like maybe avatars or large language models can be used in those settings. Gathering evidence from the real world, so monitoring how patients respond to treatments in the real world, and then using that to bring back to inform your drug development. Better predicting clinical success. Using really a combination of molecular data, cell-based data, clinical data, and then real-world data. And increasingly, much more personalized precision medicine as well. So not necessarily looking for like a single drug that's going to cover an entire patient population, but much more looking at smaller stratifications of very, very targeted therapeutics. And I quite like this quote, really. AI will not replace drug hunters, but drug hunters who don't use AI will be replaced by those who do. So, basically, it's not that we're doing it for fun or something. It's kind of imperative that you do this. Click. And in the environment that we're in, we have to be a little bit careful, of course. We're innovating in a world where there's challenges in terms of perception. There's lack of consensus sometimes. There's a lot of scaremongering about risk and things like that. We operate in a very, very highly regulated environment, which can be pretty challenging. I think the temptation is to over-regulate. So I spend a lot of my time trying to dispel some of the fears that I think are kind of less plausible, and then focus in on risks that we might face that are actually kind of more realistic to what we're actually doing. So we try and ignore the sensationalism, work with the legislation, and that means meeting with the regulatory agencies and co-creating legislation along with them. It's kind of it's not what you might imagine yourself doing after researching in machine learning, but it's a really important area as well. So I'm sure everyone's come across the acronym FAIR before, so findable, accessible, interoperable, and reusable. That's a principle that we're following very strongly. And essentially, we collected data together across the company from a vast array of different sources. And so we have a big program going on. The goal is to make all of our R&D data AI ready, basically, and fair by the end of next year. And so that means, that's a huge amount of work of ingesting, going through privacy impact assessments, harmonizing the data, making sure it really is interoperable. And so, that's the sort of, again, maybe less sort of like fancy side of things, but it's no coincidence, I think, that our data office is much larger than my AI team. So this is a huge piece of what we do and really underpins everything. So a little bit about my little slice of the world. So I run this team called the Center for AI. We're global, so we have people in Cambridge. We actually have somebody in Macclesfield as well, in the Macclesfield office, so I'm going to go down and see them this afternoon. And Gothenburg in Sweden, increasingly in Barcelona, which is one of our biggest growing sites. Gaithersburg, which is just near Washington, D.C., so that's sort of biotech central in the States. And Mississauga, which is just on the edge of Toronto. So we're not that big. We're like 30 people, so it's a few people here and there, but definitely been growing. And our sort of global footprint matches the global footprint of our clinical teams, and that's a sort of a strategic placement to have people who can work closely with those clinical experts and the wet lab scientists as well. So the picture on the top right, that's the new building called the DISC, so the Discovery Centre. That's our new Cambridge office that opened last year, and it's very fancy if you get to see it. So it's this kind of architectural and all glass everywhere, and so that's nice. We always evaluate whether we should build things ourselves or whether we should do things by partnering. I think this is, you were mentioning about having worked in different industries. This is one thing that sort of surprised me a little bit when I came into the pharma industry, having been in the tech industry, because in the tech industry, partnering was a weird sort of non-concept. You just did everything yourself, and that was it. But there's definitely a philosophy of is it always the right thing to build something yourself, or could you find a partner who could help you accelerate with that? So we have some key key partnerships, both with industry and academia. So we're working with Mila, we're working with Cambridge University via the CCAIM. We've also had a relationship going on for the last couple of years with Benevolent AI, using their knowledge graph approaches. We've been working with NVIDIA, particularly using their BioNemo platform recently. And Ion Labs is based in Israel. That's actually a sort of an incubator for startups. It's a cross-industry funded, so it's us plus Pfizer and Merck and Amazon are all involved in that. And that's brought about sort of, 5 or 6 startups that have been funded through that, and we're partnering with a couple of them. So, some of you may or may not be familiar with how drug development works. You probably know that it's takes a long time. And that continues to be true. So, this kind of gives you this is the sort of classic funnel that you might know about. The whole thing can be anything from 15 to 20 years. And that's really going from selecting which target you're going to go after, usually a gene or something like that, through preclinical screening molecules, going into, going through in vitro testing into in vivo, and then into first in human trials, where you maybe still have a few candidates, but those, as you go through phase 1, phase 2, they get down selected. And the probabilities of success, I think that's the sort of the last row. I mean, you can see, so, as you're going through, I'm not sure if you're supposed to multiply these probabilities or not, but essentially, like, the vast majority of things fail. That's just the fact of things. And that's, of course, true whether your drug is designed by traditional methods or whether you've used AI or machine learning to do it. So I think, one of the things we keep hearing at the moment is there hasn't been a machine learning-based drug on the market yet. And as well, like, how long has machine learning been around for, and how long is this pipeline, and what's the probability of success? So that kind of tells you why that hasn't happened yet. But, pretty soon, I think you'll see a flood of it, really. So this is sort of just a simplified version, because I'm going to use this a couple of times. So again, we have these we have different ways of tackling various parts of the life cycle, and we basically have data that we can use at every single stage. So the way we think about it is that if all we did was use AI to design molecules, then that wouldn't really help things that much, because we'd be sort of just feeding more things into the top of the funnel. But then if you can't run your clinical trials any faster, you're not actually going to get anywhere. So we have a portfolio of projects that covers the entire drug development process. And, sometimes it can be, small things that could impact a phase 3 clinical trial can make such a huge difference that can sort of effectively justify our entire existence, really. So we make sure that we work with our partners in the other expert functions and in the therapy areas to really understand their needs and work right across the pipeline. So we do quite a few different things using all sorts of different modalities of data. So I'm just going to do a couple of deep dives into, or deeper dives, not that deep, but you feel free to ask me more about these things as well. So I'm going to cover 3 different areas. One is in discovery, using, so this is around molecular design. Again, in discovery, looking at some of the graph-based approaches, and then in the clinical setting, looking at some of the biomarkers that we've been developing. So again, this is sort of, really early on. This is before you get to, before you even get to testing anywhere near a human. So this project is around antibody engineering. This is a really big effort within the company, involving multiple different parts. So it's led principally by one of the other expert functions, which is called biologics engineering. And then we're effectively the AI team that works inside that, but there's also some data scientists and IT folks who work in there as well. Because antibodies can be used both in biopharma but also in oncology. We have some oncology people working there as well. And biopharmaceuticals development is basically, so that's the team that will take your optimized molecule and then work out how to scale it up into something that can actually be manufactured. So this photo, I think it's actually a year old, and the team is about 90 people now, so it's a huge effort. And so again, data is key to this. So what is the data and how are we doing it? Well, the first thing, the start of this, it actually came out of a collaboration with the LMB, which is the Lab for Molecular Biology in Cambridge. The building is right next to the disk. You can see it out of the window. And it was came out of a postdoc project there where they effectively repurposed the sequencing machine to be able to do screening of antibodies on the flow cells of the device itself. That was published earlier this year in Nature, so pretty big deal, and they're forming a company out of that as well. So we've been partnering with them, and that's the sort of critical data piece, because that's capable of screening hundreds of thousands or even millions of variants of a single molecule in a single run. So we get vast amounts of data coming out of that. And that obviously is great news for machine learning. And the ultimate goal really is not to be using that forever. The ultimate goal is to be getting towards what we call de novo drug design. And in de novo drug design, you're really looking to say, well, okay, here's my target. Maybe I know where to try and hit that target or not. But let's start with a very basic scaffold, some kind of an antibody or something like that, that possibly doesn't bind in any way to that target, and then use machine learning to do the rest. Really, that's the ultimate goal of where you want to get to. In the meantime, we're nowhere near that. So in the meantime, what we can do is we can start with what we call a parental molecule, which is something that weakly binds, and then optimize around that. Use the deep screening platform to basically test many things, and then operate in cycles where we update our models and then test again. So that process can not only get us to faster sort of candidate models, it can also potentially open up increased diversity. So what that means is you can maybe find more different kinds of molecule that hit the same target. If you do traditional rational design methods or directed evolution or something like that, you're unlikely to find a lot of diversity. Typically, you find one good kind of molecule. Then the other kind of diversity is around being able to hit targets that were very difficult to do using traditional methods, and there's all sorts of reasons why that might be the case. But effectively being able to sort of circumvent a lot of lab work allows you to perhaps get towards targets that you might traditionally call undruggable. So that's all quite abstract. In pictures, in the biology, this is sort of what it looks like. So the antigen is your target protein. This is the thing that you're usually trying to bind to inhibit activity. That's the sort of the most common use case. And you can see the antibody. I don't know if I got a laser. Oh, no. What did I do there? Don't do that. Yeah, I don't know if there's a laser on that. I'll try and point. So the position on the antigen where the binding occurs is called the epitope, and then on the antibody you have these regions on the antibody which are called CDRs, so complementarity determining regions. So that's where it says CDR loops. So that's basically where all the magic happens on an antibody. You can kind of think of it as sort of quite a rigid structure with this sort of like floppy loop area on it. And it's those loops that you end up modifying in order to try and bind to the target. And the part of the antibody that binds to the epitope is called the paratope. So the paratope is on the antibody, the epitope is on the antigen, and you're effectively trying to predict the binding, paratope to epitope binding. So in sort of a really simple picture, your input is your parental protein sequence and the regions that you're editing. So these will be those CDRs, the complementarity determining regions. You might pick to only edit one or two of those, or you might choose to edit them all. And then you generate many designs using your favorite machine learning methods. Actually synthesize those molecules, so we have to go away and get the DNA synthesized, and then you test them using your high throughput affinity scores, and then repeat the cycle basically. So using your updated models for the next time around. So what are the components of this? So one of the first is representation learning. So we have models that essentially understand something about proteins, and this is similar to AlphaFold and ESM and things like that. If you've heard of these models, if you've been in this field. So again, what's the data here? Most of the models are actually trained simply on large protein databases. I keep meaning to try and find updated figures for this because that ends at 2022, but you can kind of see the trajectory. It's so the number of unique proteins in UniProt is, I don't know, it's probably 1 billion by now or close to. We tend to use antibody sequences because we're working with antibodies, so it makes sense to do that. And there are a couple of pretty large databases there. So the OAS, is everything okay? Simple, okay.
Simple if you know that.
Yeah. Thank you. So the OAS, which that comes from Charlotte Dean's group in the Oxford Protein Informatics group, so OXPIG. And that has over 1 billion sequences which come from a large number of studies. Primarily those are human and mouse antibodies. So it's kind of important where the antibodies come from, because there's quite a lot of specificity between species, and that actually becomes important when you're trying to develop a drug, because one of the things you need to be able to do, sadly we haven't managed to get rid of doing animal trials as yet, but that's another goal that we'd like to get to. But the reality is, at the moment, if you want to get a drug on the market, it has to go through animal testing before you can test in humans, and that means that whatever molecule you produce has to work in both your animal model and in humans. So we have to have cross-reactivity basically between species. Another large data set that we've been using is this thing called the Patented Antibody Database, PAD. It's smaller than OAS, but the nice thing about that, these are all molecules that have been patented, oh, it's hard to say, by various different companies and institutions around the world. So in some sense, these are drug-like molecules. So whereas the OAS is a bit more like a sift of everything that nature provides, PAD is a bit more like what humans have been doing to trying to modify these molecules. So when you're trying to create a drug-like molecule, maybe if you just relied on searching the evolutionary space, you might not get what you're looking for. So we have a few different models. This is one that we've been using lately called self-pad. Essentially, so it's using that PAD database, and it's using a contrastive learning approach to learn it. So effectively, we have pairings in that database which say so with antibodies you have basically a heavy chain and a light chain, which sort of form this Y shape together. The database holds the heavy chains and light chains separately. So what we did is we set up a task where we basically paired them together, and they were either correctly paired or incorrectly paired. So our contrastive loss there is then between correctly and incorrectly paired sequences. And that seemed to work quite well. I think we also had a typical masked language, MLM loss in there as well. And that will be, so that's going to be appearing in ICML this year, so that's some published work. And I wouldn't rely too much on I don't even know if this is UMAP or TSNE or whatever it is. I don't tend to read too much into these, other than that, if you can't get nice plots using one of these methods, it's an indication that your model hasn't learned anything. Just because you get nice plots out doesn't necessarily mean it's any good. So it's sort of a necessary but not sufficient condition that your model's working well. But anyway, this basically shows that the model can easily separate between the species within the data set, and you can see that in the OAS there's a much wider range of different species that are involved in there. You might be wondering why alpacas are in there. I think it's quite clear why you might have humans and mice and pigs as well, because those are all animal models that are used in testing. Alpacas, because they have a very particular type of antibody that turns out to be really useful. I think there are some other ones which you might not realize. That I think sharks have a very particular kind of antibody as well. So essentially, what does the representation learning do? That gives you a way of, getting your antibodies into a vector space. What can you actually do with that? So again, this is sort of a cartoon of what we're trying to do. So, you take your antibody. We have the amino acid sequence for that. You sometimes also have, you'll have other features about the protein as well, and typically you definitely have the same for the antigen as well. So this is your target protein. You sometimes will have an antibody-antigen complex. So if you have a parental molecule that binds, we can try and crystallize that, and then we'll have a complex structure of those two things bound together. That's excellent information if you have it. Tells you where your epitope and paratope are, and allows you to sort of have a look at what the binding interface looks like. And effectively, we're feeding those all into some kind of big machine learning model, and then trying to predict actually many different things from this. So it ends up being quite a large multitask learning problem. So the first thing is binding affinity. You want to know how strongly it binds. But then specificity is also really important, because you can sort of hack binding affinity by just making the surface of a protein really sticky. And then it just sticks to everything, and is not very useful, because yes, it will stick to your antigen, but it will also stick to everything else, including itself, which is really not helpful. So specificity is just as important as affinity, really. I mentioned about species cross-reactivity. That's another thing that we try and test for as early as possible. So those are sort of pharmacological effects. There's also a notion of what we call developability, and so I mentioned earlier about the biopharmaceuticals development team, whose job it is to take candidate molecules and scale them up for development. And they will look at things like, are there any sequence liabilities? So those will be like particular amino acids that are somewhere in the chain that might cause a problem, such as toxicity or induce an immune reaction in the body. Then they look at sort of aspects of the protein itself, so protein stability, yield, which is basically when you come to manufacturing, how much actual protein substrate do you get during the manufacturing process? And I mentioned the immunogenicity as well. So it's pretty hard to get all of this right. Ultimately, you'd like to do this kind of one shot, so that you have a single model that predicts everything. At the moment, we kind of do this as a multi-stage process. So we look at affinity and specificity first, and then we look at cross-reactivity next, and then we have a chain of models that all do the various different developability properties. So the picture shows one model. This is actually kind of more like about 10 or something at the moment that operate in a big pipeline. But essentially what we're aiming to do is, when we generate the library, so the big sets of candidate proteins in the first place, we'd like to get to the stage where we can predict all of these things ahead of time, so that you're producing only good molecules, basically. And of course, everyone talks about Gen AI at the moment, so I had to point out, some people I have to point this out to, that this is generative, it's a generative model, so it falls under the umbrella of generative AI. Not for this audience, sorry. So how do we go about the design process? Again, so this is another little cartoon to show you what we're trying to do. So here we have the antibody on the left. We have the three CDRs, so the complementarity determining regions. You have your antigen, and in the blue of the antigen is the epitope. And essentially you're trying to generate from scratch what these CDR loops are going to be like in order to bind. There's various different ways that you can do this. You can do some really simple things, actually. And in a sense, the way I like to think of it is that if you compare two models together as well, so you can have a generator and then some kind of oracle or some kind of discriminative model whose job it is to rank the generated candidates. And if you had a perfect oracle, one that could accurately rank everything that's generated, then your generative model wouldn't have to be very good at all, because you could generate billions of things using very, very basic search methods. You wouldn't even necessarily need to have machine learning in there at all. And vice versa, if your generator was perfect, then you wouldn't really need an oracle. You wouldn't need that model at all. The reality is that neither are that good. We know that the generative models, I think, the way I like to think of it is biology is way more complex than natural language or computer vision or anything like that. So, we've made huge strides in generative models in those areas. I think in the protein world and we're only really just scratching the surface at the moment. So we can't just rely on the generative models alone. And the discriminative models, the oracles that we build on top of the high throughput sequencing data, are also not that mature yet. So we rely on both still. So we generate things and then re-rank them. And we sometimes use ensembles of models to do the re-ranking. I think, and whatever, you can have an encoder-decoder structure or whatever you like. It doesn't really necessarily matter what your generative model is. You might have a diffusion model or something like that. But, those are the essential ingredients, really. And, how might you go about actually designing one of those libraries to be tested? So again, you're using your generative model. We always seek to have a diverse set of sample sequences, basically because machine learning models are very good at finding local optima. Then if you just sort of had tons and tons of sequences that were all around a single local optimum, then if one was wrong, then the likelihood is they're all wrong. So we try and encourage diversity when we put together these libraries as well. We include an implicit bias that we don't let the models go too wild. So this is basically trying to stay nearer to the parental sequence rather than just, going completely crazy. And partly that's around not really trusting the models, especially away from the training data. But also it's because typically we know that the parental sequence has some sort of good properties, that it's less likely to be, toxic or whatever. And if you go miles away, there's much more likelihood that that's going to happen. So a really simple way of doing this is the first thing we did was some sort of ancestral sampling. I think we've iterated on this a bit since then, but your inputs look like something like this. You have an amino acid sequence of some length. Now here we're just saying we're doing things with a fixed length. Ideally, you would want to be able to say, because these loop regions can actually be a variable length, so you might want to say, oh, you could add in or subtract a few amino acids, but the simplest version of this just assumes it's, say, 50 or 100 amino acids long. And then you basically, take your embedding model and just predict softmax scores at each single residue. And then we have some parameter which is defining how much exploration you'd like to do. And so using that exploration parameter, you basically sampling a number of mutations that you're going to make using a Poisson distribution, and then you're effectively picking random locations along the protein sequence. So you pick those K mutations along the sequence, and your probability of choosing a location to mutate is just going to be the entropy of the softmax at that single residue location. So this effectively says if the model believes that the certain residues are more variable in what it's seen in the training data, then you're more likely to mutate at those places. And that's basically a sort of well, I guess either an evolutionary bias, or depending if this is using that PAD data set, it's more like a human bias on where things are mutated. But there are parts of the CDR where it will make more sense to mutate than others. And hopefully the model has kind of captured that within those softmax probabilities. And then at that point, given that you found those locations where you want to mutate, then you're just drawing from a discrete distribution where now the probabilities are the softmax of that particular location. So, most of the time it would choose, it could just choose the same amino acid that was there at all, because we don't remove that. But depending on the sort of how peaky that distribution is on that single residue location, you might get sort of more variability around there. So at each location that you choose to mutate at, you're still going to get somewhat sensible mutations. It's not going to give out ones that will totally sort of destroy the protein and make it not fold anymore or anything like that. Of course, like one of the weaknesses of an approach like this is it's not making use of the context of the changes that are being made. So you're making use of the context as you're choosing where to mutate and which mutations to make. But as soon as you made a mutation, like a really trivial example of where this goes wrong is if you put a cysteine in one place and then a cysteine in another places. Pairs of cysteines like to make these things called cysteine bridges, which is like a salt bridge, which can sort of cause the loop to fold in on itself. So this is the sort of thing that you would have to, if you would kind of have to post-process to remove things like that using a naive method like this. So some results. So this was the first machine learning library that we produced, and this was last year. Using a real target, and we were basically going up against traditional protein engineers using rational design. So for the rational design, that's effectively where you're using Rosetta and tools like that to manually change and look at the energy landscape. So using free energy perturbation and other computational methods to look at the changes of single amino acid positions. It's typically very, very hard to do both the heavy and the light chain at the same time. So with the rational design team, they did two separate libraries, one looking at the light chain, one looking at the heavy chain. We didn't have that restriction for the machine learning libraries, who were able to edit both at once. We picked to use 5 out of the 6 CDRs, so 5 out of the 6 regions. I don't exactly remember why one was fixed, but that's kind of by the by. And the bar chart is showing the number of sequences that were had a higher binding affinity than the parental sequence. So you can see that, but using the machine learning method, almost everything that was selected ended up being a better binder than the parental sequence. Whereas using the rational design methods, it was a much smaller proportion. And this was kind of amazing because you know, if I'm honest, the models are still pretty naive. You know, this is, you know, using just sort of large transformer models, but nothing fancy at all. And quite simple predictive models, just based on large amounts of data. I think, you know, we've been developing since then, and we can you know, you can definitely dream up much more interesting kinds of model. But it sort of shows that even with relatively simple things, you can already get quite a long way. Now, of course, this, just because you have lots of things that are better than parental sequence, is only the first part of the story. You still have to do confirmatory screening. You have to do all of those other processes. This is a long way from a molecule being, you know, ready to make into a drug, but it's a really nice start. And this was another one. So this was quite a challenging target that we were asked to work on. In this case, there wasn't a rational design library at all. It was deemed as too difficult to work with, and it was essentially, well, we're not going to bother with this project. It's too difficult. Why don't you have a go with your machine learning methods if you want to? You know, again, here you can see, so that the plot is basically showing, so intensity is the intensity on the screening platform, because this is using, you know, as I said, it's using sort of a next generation sequencing, an NGS approach, where you're actually looking as like fluorescent intensities on the flow cell. The intensity here is basically a correlate for the binding affinity. It's not perfect, but it's a really good indicator. You can basically see the so I think this is the on and off binding, and you can see that the library produced a huge set of sequences that were all able to bind to a much greater degree than the parental sequence. So at the x-axis is time. So this is basically showing what you want to, you see basically the on and the off event, so the binding and release of the target. So that's your sort of KD on and KD off, if you've ever come across those numbers before.
How much time does it take at the beginning of
I don't know the answer to that. Yeah. I'm not sure. I'm not a biologist myself, so I'm going to pass on that one. But yeah, maybe we should. I should ask someone to give me an axis with the x-axis labeled. I always tell people to label their axes, so I'm a criminal here. I mean, essentially, you know, you want to. You know, you typically see these curves of sort of binding and release, and I think you know it's. I think you know you typically get a sort of a longer release period afterwards, but that you know they will look at the peak binding energy, but they will also look at you know, they'll look at the slopes of the curves, and that sort of also helps determine the properties of the chemical of the reaction as well. I mean, I'm going to move on to a different example now. So I don't know if I'm happy to take questions on this at this point, if that helps. People are sort of interested. Yeah, so basically, you know, I think what it is that you're changing the temperature as you go through over time, and then at some temperature binding occurs, and then as it gets hotter, it releases again, something like that. You're asking me too many questions about this plot that I don't know the answer to. I was kind of meaning more general questions about the last section. Yeah.
I have a question about the antibodies. So you were mentioning about a model that does contrast, right? Like it has two encoders and it's contrasting between things that like alpha and beta chains that bind from alpha and beta chains that don't bind.
Yeah.
But can you just? I've seen examples where you can just pick a random alpha chain and a random beta chain. Okay, not completely at random. If you do some educated guesses, these can stick together, and you can form this sort of chimera, let's say.
Yeah. So, no, that's an interesting point. In your contrastive learning, there could be negative examples that are actually positives in there. I mean, I think that's kind of always the case in contrastive learning. You know, it can be quite difficult to select negative examples. And sometimes actually your negatives are just too easy because they're things that obviously won't match up. And the kind of the interesting negatives really are the ones that are somewhat in the neighborhood, but you know, somewhat plausible, but they're not really. And that's what helps the model. They're the really discriminative ones.
How much are you biasing when you I understand you need to bias because you're looking to engineer stuff, but how much do you think it's affect?
Biasing in what sense?
So there is so you're making an assumption there like certain alphas and beta chains can't go together where in fact they can't go. You can't observe them in nature. Right? So this is like you're making a simplification of
Yes, but the thing is, those ones that can actually go together in nature are very, very unlikely to be ones that would make good drugs anyway. Because those sort of chimeric ones and things like that, the likelihood is that they're just going to flag up for, you know, that will be something that the body would attack. It's something that doesn't look human-like, basically. So in a sense, when I said about how we generate libraries and we have this prior about staying quite close to the parental sequence, I would say that holds as well for the whole structure of the antibody itself. If you know, so some of there's some other work in protein design, for example, you know, the stuff that comes out of David Baker's lab in University of Washington, where they're dreaming up all sorts of things and they're making like little protein motors and, you know, kind of, you know, things that nature would never ever produce, but like are really interesting sort of, you know, nano machines and things like that, which is amazing, but none of those could be used as drugs, right? Because the body would just, I mean, you get antibodies that attack antibodies, so they're anti-antibodies basically, and you have to avoid those. So if the molecule you produce doesn't look like it came from doesn't look like another human antibody molecule, it will just get attacked by antibodies very quickly. Great, okay. How am I doing for time? Half an hour, okay. Well, that was the biggest section, and so these two I can kind of run through a little bit quicker and then give you some more time for questions or get you into the break earlier maybe. So again, this is preclinical, and this is work really that's like kind of applicable to the entire sort of preclinical phase. So this is around using graph-based methods, and you can use graph-based methods to do various different things. So you can use it to do target identification. You can use it to do, you know, to help with some of your lead identification and prediction of tox and things like that as well. So effectively, I think of knowledge graphs as being a bit of like a Swiss Army knife that can be sort of applied to many different things. It can actually also be used in some of the later phases as well. to do things like drug repurposing. And actually, I think one of the use cases covered here is around that. So it's pretty natural to think of biological systems being represented as a graph. So here we have, a very small but sort of, kind of example of what you might have in one of these graphs. So you have biological processes, you have cell types, you have tissue types, you have anatomy, parts of the body, you have genes, compounds, diseases, pathways, phenotypes, that kind of thing. And these are all, triples, so you have this gene upregulates this other gene, or this compound binds to this target, etc. So we have various different knowledge graphs that we've been curating. We have one in particular, which is very, very large with billions of data points in there. And the sources of data are quite varied. So that's, some of the pharmacology stuff. So there's, wet lab experimentation, chemistry, biomarkers, genomic data, clinical trial data, other kinds of omic data, so transcriptomics, proteomics, and even, sort of things that are coming from the literature, so scanning papers and abstracts, images, which can be, MRI or echo or whatever, cardiac images, electronic health records, patents, really anything. You can sort of represent anything. Of course, it's only as good as the curation techniques. So this, harking back to the sort of the fair data slide and harmonization, it's not just sort of a dumping ground. You have to be quite careful about, how you put things in, how you create it. So we have a lot of subject matter experts who take part in this process. So this one that we call the biological insights knowledge graph. Roughly, I'm not sure when this was, but 11 million nodes, 850 million edges, 5.5 million compounds, 110,000 diseases. So, pretty large database. It's, of course, is not 100% reliable, as with any knowledge graph. And so anything that any predictions or anything that or any information that you extract from the graph, you have to of course validate. But sort of a resource that no one human or group of humans can keep in their head at one time for sure. So the use case here is around indication expansion. So this is when you're trying to find new uses for an approved drug that's outside of the scope of its original indication. This is a really kind of attractive thing to do because, typically you will have gone through all of that preclinical phase, and maybe some of the early clinical work as well. For example, if you've already gone through animal testing, you wouldn't have to repeat that because there's nothing about. So when you do the early clinical work, and even the phase 1 is really about safety and toxicity, and is nothing to do with the efficacy of the drug. That comes in the later clinical trials. So if you then repurpose the drug to a different indication, you don't have to do that safety and toxicity again because it's already been proven. So that's a massive saving if you can do it. So the traditional method for doing this is some kind of manual literature search, an internal data review of all those different data sources, some kind of manual ranking of options, and then sort of validation, clinical validation, wet lab validation usually. Essentially, this is the automated or semi-automated way of trying to do the same thing. So using the BIKG, or it doesn't have to be, it could be another knowledge graph. We have a graph machine learning inference engine that's been built on top of that, and that will produce the ranked list of novel indications. But not only that, you can use the graph to then introspect on it to produce what we call mechanism of action importance maps. So this is trying to get at sort of the biological understanding of why that indication might be a good target for that drug. And then it goes to expert review, and you might go through the same procedure of doing some sort of wet lab validation or something to prove it out. I think what's the easiest way to say this? We're basically using graph attention networks, which you're probably pretty familiar with. It's essentially basically just using node embeddings plus some topological features on there. So it's really mainly using the connectivity of the graph, plus some information on the edge. On the nodes. There's no edge weights in these graphs, so we can't make use of that. But at that point, and we're using this typically in a transductive setting. And then when we see one of these indications come up, we also like to try and have some notion of uncertainty along with them. The biologists who use this tool always ask for that. Basically, they want to understand, like, oh, okay, well, you've ranked this thing above this other one. How confident are you in those rankings? It's not totally trivial to get uncertainties out of GNN, so we've been using MC dropout, which is sort of simple but reasonably effective, and that allows us to have some notion of uncertainty quantification. And for the explanations, I haven't got a slide in here for this, but the explanations are effectively just looking at the attention weights in the graph attention network, and that allows you to then basically so once you've selected your novel indication and you have the molecule that you're interested in, you just follow through the graph and you look at the attention weights along all of the paths that would take you to that, and then so you can weight the different trajectories according to the attention weights, and that basically can sort of plot the most likely routes through that graph using that. And that it kind of gives a picture that there may be more than one possible way in which that drug influences that particular target, because that's often the truth. It's pretty difficult to actually validate that those attention scores are correct. I mean, I think this is a bit of a maybe a bit of an unsolved problem with these things, that quite often the ground truth for something like a causal network within a biological process doesn't really exist. So if people here are interested in working on something, I think that would be a really interesting place to look at, sort of trying to get a bit of a systems biology, trying to understanding of what's going on. But yeah, I think these tools are intended to be useful rather than giving exact answers, I would say. Okay, so the last example And this is just to say, doing something completely different, and now for something completely different, basically. So this is looking at using computer vision, and this computer vision can be used, right across the different clinical phases. I think this work in particular is mainly aimed at around phase 2, so the late early clinical phases. We look at many different modalities. So we have, so I think that's cardiac MRI in the bottom right. That's a retinal image. That looks like some histology staining. And then we have, just images of, people's skin and things like that. We have a few different things that we're trying to do. One is biomarkers. So biomarkers are basically indications of whether whether the medicine's working or not. Quite often, you'll have your sort of primary endpoints in a clinical trial, which will be things like recovery from symptoms or not dying. Those are the main things that you're looking at. But then you have biomarkers, which are additional things which you can measure along the way, and you would hope that those biomarkers somehow correlate. They either correlate to the symptoms or to the actual underlying disease itself. And you can use the biomarkers in another way, which is to sort of pre-segment patient populations when you're actually designing your trial in the first place. The other thing that we do quite a lot of is quality control. This is sort of something which is kind of quite an easy win for machine learning in a way, because in quality control, you're not trying to assess whether somebody has a disease or not. You're just trying to say, was the image that was captured a good image for that purpose or not? And it turns out that that's actually a really important problem. For example, in echocardiography, it's quite hard to get the image capture right, and I've seen sort of numbers that anything between like 25% and a third of all image captures fail, and they fail for multiple reasons, but quite often it's because if you think about what's going on, it's typically it won't be a specialist that's doing it. It probably isn't even a nurse. It's probably like a technician who and it's one of those jobs that it's quite high turnover. It's a hard job to do. You have to do many of these things during the day, and they just it's just a sort of a fact of life that you get a lot of these failures of images. And if you have that happen and you don't, it's not detected at the time of capture, that the patient goes home, the likelihood that you're going to get them back in to capture another image is extremely low. So you've basically lost data there, but it's even worse than that, because nowadays with clinical trials you have to you have to specify ahead of time who your patient population is, and every patient has to count, and it has to be included in your data. So if that data is missing, it's just missing from your data. You can't get another patient and add them in. Something as simple as having an online system that works in the clinic itself, in the hospital, and just flags up, no, please retake this image, is actually quite a big deal. So that's something we've been working on as well. So a quote from Neil Lawrence here. The real danger is that computers don't anthropomorphize. They'll make decisions in isolation from us without our supervision, because they can't communicate with us truly and deeply with us. The reason I think this is sort of a well-known thing, that basically if you train a discriminative computer vision model and you're not careful about it, then it will easily pick up features in the image which are nothing to do with the actual discrimination task itself. And there's some really classical, errors. I think there was something about, tanks and clouds or something like that. I can't remember these really, really old examples. But it still happens today, and we have to be really, really careful about that. So we've been looking at this using a method to basically use counterexamples to interrogate the latent space to understand what the model is using to do its classification. So this was sort of like loosely based on the activation maximization framework that was in the original Google Deep Dream. And the idea there was you basically run an optimizer on your input, the image itself, to maximize an activation, and then you get these like really weird sort of dreamlike pictures that come out of it. And in some respect, that tells you something about what the model's representations, space looks like, and some of the things that are in there. I mean, obviously, this isn't terribly useful as it is, but you can see that there's sort of, the model's obviously learned things about, I don't know, birds and wheels, and those are all things that are appearing in those images. So if you can sort of direct that in some way, it could be quite useful. So we were looking into this for some retinal images, and we started off doing it with GANs. This is a few years ago, I think. We were looking at this Starlex method that had been published, but actually this was really not working well. So this was producing basically things in the image which these were kind of hallucinations, really. They were sort of like artifacts. It was picking out artifacts in the image that were nothing to do with the actual disease that we're looking at. So then we kind of moved over to using diffusion models to do the same thing. And, obviously, like, there's hundreds and hundreds of different methods with diffusion models. But, essentially, I guess you're all familiar with this. I'm sure you're, but effectively you're starting from some random noise and trying to diffuse back to a full image, and you can do this for segments of images as well as for the whole image. So you can do this kind of in-painting tasks and things like that. And so here, this is a very basic method here, but essentially, you're taking some kind of image, some sort of image where the person is showing the sort of skin disease or not showing the skin disease. You're basically blurring the image and then optimizing the input to maximize the counterfactual class. So either you start from a healthy person's skin and then you try and diffuse back to unhealthy, or vice versa. And then you can kind of, I think in this example we had a healthy skin, so you're diffusing to the disease state, and we just did a bit of blending back in with the original image just to make it look more realistic, but effectively, then it's sort of showing what it's picking out as the elements of the image that the sort of the model believes to be the indicative of the disease, and interestingly, these sort of areas around here, I think that's the sort of key area in this image, is where you get a lot of this. A lot of the information happening. So it seems like the model's kind of correctly learned the morphology of this particular disease. And then we can do the same thing with retinal images. So it's exactly the same approach, different modality. And here you were able to pick out different sort of parts of sort of different phenotypic parts that are coming out. So the vasculature and the microaneurysms and the mottling, they're all sort of different things that might show up when you have. So I think these are images from chronic kidney disease. So when you have kidney disease, it actually affects what's going on in your. You can see it by looking at retinal images. And another one, this is from sclerosis. So once again, this is just again showing that the model's coming up with sort of plausible explanations, but in yet another modality, which is sort of totally different. And again, this is something you can sort of do it controllably as well. So in these images, that it's actually, rather than being a binary classification problem, they have like a severity scoring system, and we can have the model sort of pick out different severity scores and show those as well. How are we doing? I feel like, oops. All right, okay. I think I want to wrap up now. Yeah, so effectively, if we just rely on the classifiers alone, a lot of this is about buying the trust of the clinicians so that we can, use these models in reality, and they want to see these kinds of explanations come out. So this is sort of an important part of getting buy-in. Yeah, so that is it. Whistle-stop tour. And so QR code just takes you to the AstraZeneca website. It's not that interesting. Well, it's sort of the data science and AI area. And thank you for your attention. I think we have a few minutes for questions.